Great, let's get started. Jenny, do you want to welcome everyone?
Yeah, sure. Definitely. Welcome everyone to our fireside chat with Nick Hertz, founder of Mitochinen. I'm Jenny Gianci, program director of the UCSF Catalyst program and the director of the UCSF Launchpad. Our Catalyst program is our on-campus translational accelerator, and the Launchpad supports student, postdoc, and trainee entrepreneurship. So it's a pleasure to be here with Nick and Zach today and bring Nick's story to our audience. I'm sure many are curious about his experience starting Mitochinen out of UCSF. I want to thank Zach Collins of MBC BioLabs for reaching out to us and to put on this event. And of course to Nick for his willingness to share his experience. It should be enlightening and hopefully even inspires some of you to take the leap towards entrepreneurship as well. So thank you all for joining us today, and I will turn it over to Zach.
Thanks so much, Jenny, and thanks, Nick, for your time, and thanks everybody for attending. Just as a super quick intro on myself, my name is Zach Collins. I'm the head of academic alliances at MBC BioLabs. I also help out at our venture fund, Mission Biocapital, as an associate. The super coolest thing about working at MBC BioLabs is getting to work with cool scientists like Nick every day. We're building biotech startups, one bench at a time in our labs. Right now, MBC BioLabs is host to 97 companies across the Bay Area in San Francisco and San Carlos across our 3 locations. And we really want to try and help early stage startups get started, raise money with that key data that they can get at our labs. So far, companies in our labs have now raised about $10 billion here in the Bay Area and launched dozens of clinical programs. Nick is one of the most exciting stories to come out of MBC BioLabs, so we really thank him for taking his time to join us today. Nick is the CSO, Chief Scientific Officer of Mitochinen. Nick started his scientific journey, or at least in academia, at UCLA with a BS in biochemistry. He did his PhD at UCSF in the Chemistry and Chemical Biology program, where he worked with Kevon Shokat and Homo Burlingame. His work at UCSF culminated in the founding manuscript behind Mitochinen, which revealed a new way to get after PINK1, a key disease-causing gene in Parkinson's, and actually unlock it as a therapeutic target where others had failed to do so. After leaving UCSF, Nick was a Helen Hay Whitney Foundation Fellow in the lab of Mark Tesser-Levine at Rockefeller. While a postdoc, Nick founded Mitochinen and has since gone on to raise $18 million in venture financing and entered into a partnership with AbbVie earlier this year. Nick, thanks so much for taking the time. If you could tell us a little bit more about Mitochinen, we'd love to learn a little bit more.
Yeah, of course. And I just want to say thanks so much to Zach for all the work that we've done with you and Mission Biocapital, and how fantastic. If you guys are all thinking about starting a company, you should definitely go and work with Zach and Doug and the team at Mission Biocapital. They're fantastic to work with. They're a great founder-friendly investment firm. And so here's a little bit about Mitochinen. I'll just show 3 slides. And so let's see. So basically, there's many sufferers of neurodegenerative diseases, but there are 0 disease-modifying therapies. And so our goal is to really pioneer a disease-modifying therapeutics for a neurodegenerative disease. And so the way that we sought to do this was focusing on the human genetics, and I think that's why everyone's getting, as Zach was saying, getting excited about our approach. Is that if you look at the penetrance of genes, so basically on the bottom here is the allele frequency. So you can see the these are common mutations. You have many different people who have these mutations, but there's not a huge correlation with getting Parkinson's. Right? Sorry guys, my slides are advancing without me. And then as you move over this direction, you see these are very rare variants, but actually these have a higher risk of actually getting the disease. And so you can see the focus here is on PINK1 Parkin, sort of these genes, which are all mitochondrial associated. And we chose to focus on these genes because despite them being very, very rare, so there's not a large patient cohort to treat, but because of the fact that patients with these mutations have the disease. So we thought, you have a normal level of these proteins, and you don't get Parkinson's. You have a lower, if you have a lower level, then you get Parkinson's. So if we can bring that level back up, or even amplify the activity of the normal, gene, we can maybe prevent Parkinson's in people who don't have mutations in those genes. So that was really the impetus for the company. And I started working on PINK1 back in about 2008, and it was only discovered in about 2004. So we've really been sort of paving the road as we go down and develop the therapeutics. So it's really like there's no great antibodies, there's no great tools to do it. So we really have to do a lot of that work as we go along. And so just overall, what does PINK1 do? So, and this has been work that actually resulted in a breakthrough prize last year for Richard Ewell, who elucidated a lot of the biology behind PINK1. And so it's a really quick pathway to engage. So when mitochondria are healthy, PINK1 is actually taken into the mitochondria and then degraded. But when the mitochondria are damaged, PINK1 is stabilized on the surface of the mitochondria, where it then phosphorylates substrates, including itself, resulting in the recruitment of parkin, another gene mutated in Parkinson's disease, which then ultimately results in the recruitment of autophagic machinery to those mitochondria and clearance of them by the lysosome. And so we believe that the disruption in this process results in the accumulation of damaged mitochondria in patients who actually get Parkinson's at very young ages. And so our approach is to amplify the activity of PINK1 only when it's already stabilized on mitochondria through its normal regulation. And so, we've taken this approach and now we're nearing to entering the clinic, hopefully in 2022. And so I think the panelists have questions, and so I'll just stop sharing slides now.
Thanks so much, Nick. That was great. Let's rewind really far back to the absolute beginning. How about that? Tell me a little bit about where you grew up. What was your childhood like? What got you interested in science?
Yeah, as I told you yesterday, we have a lot of stories here for science, but I'll keep it short. So I really became interested in science just actually just wanting to understand, what the universe is made out of, and what, I remember sitting there in class and thinking, what are all these materials? What are they made out of? So I focused on chemistry initially, just trying to understand where things come from. And I, as I told you yesterday, I think I have actually in my 8th grade class, we had to make a diorama of one of the organelles inside of a cell. And the diorama that I made was actually a mitochondria. And so that was when I first, became interested in mitochondria. And, people who know me know that I have a lot of energy. And so I'm also really interested in mitochondria for the, understanding where my energy comes from. And if you get, me and my mom and my grandma all together, you'll see we all have, very similar amounts of energy. So maybe that comes from our mitochondria. So I actually grew up in Southern California in Malibu. And just got really interested in science from an early age.
Super cool. And so that brought you to UCLA, where you did your degree in biochemistry. Could you tell me more about kind of how you navigated your way towards more chemical biology and that side of things, and kind of ended up at UCSF? What kind of brought you there?
Yeah, no, that's sort of a natural flow for me, where I came into UCLA and I really wanted to focus on chemistry, on organic chemistry, because I was just really good at it, And so when you're really good at something, you tend to enjoy it. And I focused in on that. I was in the chemistry honors program, and I did, many different classes. And actually, my dad was a really strong influence in this as well. And so he said, the moment I got to UCLA, he said, you have to go do research with the top lab at UCLA. Okay, fine. And so his friend was an artist, Tom Woodle, and his brother was actually a professor at UCLA in organic chemistry. And so he introduced me, and I went into his lab and I proceeded to, break things and set things on fire and generally have a fun time in lab for a couple of years. And that's where I realized that organic chemistry was not, the place for me to be. Actually, I saw it more as just that I think a lot of the organic chemistry was going to be outsourced out of the country. There wasn't really a space for being really, really creative in organic chemistry. And so that's where I switched over. So I figured that biochemistry, and it's still really challenging to do good biological experiments. So I figured that was a space where you could make a lot of headway. And so I kind of combined the two together and saying, okay, I want to understand the biology, but using these really cool chemical probes. And that was what got me into chemical biology and sort of, launched that part of my career.
Very cool. And UCSF seemed like a great place to do that too. What brought you to Keyvan's lab specifically, and how'd that end up?
Yeah, this is a fun story too. So I basically, before I even applied to grad school, I emailed all these professors. I went and researched each university, and then I emailed all these professors, and I independently went to each of their universities to talk to the professors before I even applied, to know who I wanted to apply with. So Keyvan actually tells this story once in a while too, where basically I came out and I'm still an undergrad, and I just emailed him, hey, let's talk. He said fine. So we hung out for like half an hour, and we kept talking, and I talk a lot, so we kept talking, and it went over, and he's like, well, Nick, I have to go to the seminar. You guys, shut up for a second. And so I said, okay, fine, I'll come to the seminar with you. So I went downstairs, and we were at the seminar at UCSF, and a really famous professor was, giving this talk, and get to the end of the conference, the seminar, and then the first, so Kayvon tells it from his perspective. He's like, the first hand that shot up was mine. I was like, I asked all these questions from the professors, and so I think we really got off to a good start because he figured, I knew that the questions weren't that great, but I still had the interest to ask the question. And I think that was what got me interested in working with Kayvon, was just that he had that same excitement. And so I went around to all these different universities, and it just ended up that Kayvon and I really matched up well in that sort of excitement factor of like, let's figure the science out and just keep going and push it forward. So that was how I got led to UCSF and Kayvon.
Very cool. What kind of led you to the project itself and working on Pink One and that whole pathway?
Yeah, that was the story basically. So again, I was really interested in mitochondria, and so Kayvon studies kinases, and so when I initially did a rotation in his lab, I did a lot of chemistry around kinase inhibitors in like oncology applications, and one of the molecules I made during my rotation actually ended up being used for another application for making these aurora kinase inhibitors. So that kind of turned into something on its own. But when I went to Kayvon to join his lab, he didn't look very excited. I walked in and he said, no, I'm not, I don't know, because I'd already done the rotation. He had a lot of people who wanted to join. And he told me later, this is kind of, I shouldn't give all Kayvon secrets away to people who know him probably, but he said that he likes to know that people really want to join his lab, so he kind of doesn't want to get really excited at first. So then I came in and he's like, well, I don't really know about making kinase inhibitors and blah, blah, blah. He said, try to think about something and, we'll talk later. So I said, shoot, I got to figure out something, else to study. And so, I went away and I kind of worked out, I read through his papers, and I was really interested in mass spectrometry. And so I came back and I said, I know we were talking about making kinase inhibitors, but I'm really interested in this other project. And he got really excited because he had nobody working on that stuff. So he said, oh yeah, let's identify kinase substrates. This is really great. But from his perspective, it was really challenging to do that sort of work in the bulk cell. And so he said, well, let's think about like different subcellular compartments. And I said, well, I love mitochondria. And he said, okay, well, let's figure out how to, what can we look at in mitochondria? And so I went back to Vamsi Mutha's paper on the mitocarda, which showed basically really careful subcellular isolation of mitochondria to characterize what proteins are there. And actually Pink One popped out as the only non-metabolic kinase in the mitochondria. So, and then I started reading through, and I had already proposed a couple other kinases that Kayvon shot me down on before, which was like ones that everyone studied, AMP kinase, other things. He said, no, let's find something new. And so I found Pink One. I was like, oh, this is perfect. It's like, only non-metabolic kinase in mitochondria. I like mitochondria. We can use the subcellular isolation to look at them using proteomics. And it actually has this really cool interesting disease association with Parkinson's. And so that was kind of how the project, triangulated. And also, I have a little bit of a history of some neurodegeneration in my family. So I wanted to sort of also, that was something that I was really, emotionally like interested in, just thinking about what we can make an impact on. So that was kind of how the project came together.
That's quite the Excalibur challenge for, a rotation student. I don't think I was quite so sophisticated when I was there. Jenny, I know you have a few more questions, so I'll hand it over to you.
Yeah, I guess I'm curious, what made you first interested in starting Mitochondria? Did you come to UCSF knowing you wanted to start a company?
No. My dad actually, had always kind of pushed me to the academic side of things, doing the science and really, pushing in that direction to being like a professor and this kind of thing. And, so I had a lot of respect for that. My dad was a professor for many years, and actually we have a number of famous scientists in our background. So Hertz was actually my ancestor, who was the first person to show that you could generate an electromagnetic wave through space by oscillating electric charge. So that's why a cycle per second is a Hertz. And so, it was always like, you have to live up to these, really famous scientists in your past. And, the only way to do that is by, being a professor. And so I was very much, going down that route. And so, when I applied to grad school, I told all the professors, I want to be a professor at Tier 1 research university and all these kind of things. And so it was absolutely not on my radar at all to go to a company. And so, when we got near, midway through my time at UCSF, I took this class, which I shouldn't even mention this class anymore because the guy's in jail now, but I took the idea of the IPO class back with Steve Burrow was teaching it. And so that was kind of what got me interested in starting a company, because I saw, the kind of areas they were tackling were, I don't know, it's like broader, but also more selective. And I think it was the idea of like applying the research to curing a disease that was really exciting for me. So in that class, we proposed a different company idea with Kayvon, where we were going to identify kinase substrates for all these different kinases, where there was no mutations in the kinase, but nothing was really known about what those kinases did, and there was no drugs targeting those. So actually, since now a number of companies have come out that were actually pursuing that basically that same approach. So it wasn't a horrible idea, but we didn't win the competition, and so I went back to the lab and I was working on the PINK1 activator ideas. And so Kayvon actually had proposed we should spin this out, or we should start some sort of company first. And I said, yeah, just like get it off my plate. I just want to go and be a professor. I don't want to do any of this company stuff. So don't bother me. We'll just spin it out somewhere. And so Kayvon had looked into different ways to do that. And so it was around that time I went out to the East Coast for Easter, and I was talking through the data, and we had this really cool data working with Ken Nakamura, and he showed that we could apply these compounds to primary neurons, and we could see the mitochondria would stop moving along the axon, but in the PINK1 knockout, the compound didn't have an effect. So it was at least pushing us in the direction that the compounds were selectively activating PINK1. And so I was looking at these videos, and my brother-in-law Dan DeRoulet, who's the CEO of Mitochondria now, he came over and he said, "Oh, this is really cool. What are you working on?" And so he knew more, but I talked him through it, and I talked him through a little bit about what Kayvon was thinking about how we could commercialize it. And he said, well, actually, there's another story like this too with Doug Crawford. I'll tell you in a little bit, but basically where he said, well, would you be interested in another offer? Would you be interested in raising money yourself and trying to start a company yourself? And I said, well, I hadn't thought of that. I guess that seems like it, could be a good idea. And so he went out and we tried to, we basically talked to Kayvon first, and we formed the initial company, and then went out and raised some money to get the initial ideas off the ground. But for me, the idea was always to go back to academia and go back and do a postdoc and apply to be a professor. And so it wasn't really, so we were basically launching the company while I was starting my postdoc with Mark Tessier-Levine, but with always the goal that we would sort of try to see how far we could push it, but we didn't have enough money to pay salaries and all this other stuff, so just kind of pushed on the side while we were doing our sort of day jobs.
So that's great. And what was the turning point of just having someone else run with it versus you taking it on your own?
Yeah, well, I guess that came when basically I was in Mark's lab at Rockefeller University, and then one day I was in the TC room and pipetting away, and someone came in and they said, "Oh my God, Mark started a company." I said, "Oh, what happened?" So he started his company Denali. They raised like $200 million. I was like, "Oh wow, that's really cool." Like, that's interesting, And then like a year later, oh yeah, guess what? Mark's president of Stanford. Okay, what's going on? So we, after a lot of hemming and hawing, I had this really interesting project with Mark, and so we, with my wife and 2 kids, we decided to move back across the country to California to Stanford, and still the goal of being a professor. And then got out to Stanford and pushed the projects forward, and then I saw other postdocs just like really struggling to get professor jobs, and that was the thing where. And that was probably the turning point for me. It was really just seeing, how tough it was to find that dream job, and knowing what it would take to get there, and knowing, I had this cool project that was, it was kind of near the end, but it wasn't going to be like the Nature Cell or Science paper that was going to get you, which seems to be required for getting an academic job, even though a lot of universities pay lip service to the fact that they don't look at these publications. You see the people getting hired always seem to have one of those publications, right? So I guess for me, it was that moment where I was like, oh man, how am I going to make this happen in a time frame that's going to work for me, and ended up at a university that I really want to be at, and also not disappoint my dad. And then but basically at that point, Kayvon was contacted by a large venture capital company, and they said, we're interested in your next idea. And so we ended up getting an offer from them, after a lot of negotiation, back and forth. And the idea was that we were going to launch the company with that venture capital company, and then we were going to, I was going to come in and help out and get things started, but there was going to be someone else who was going to come in as CEO, and me and Dan were going to kind of be out, and they were going to take the whole thing over, which was kind of either what we wanted or what we thought we wanted. And so it was actually then this really fortuitous moment where I met Doug Crawford, where really changed the way my life went. So it was pretty fun. We I basically we were like, okay, so we had this offer on the table. We had ostensibly the money ready to go, but we hadn't like transferred the money yet, and it wasn't in the bank yet. And so I emailed Doug via another previous MBC BioLabs 953 company. She was Becca McLaughlin. She was a PhD student at UCSF. She's now at Genentech. And so she said, yeah, talk to Doug. Doug's great. Doug will do everything. Doug will help you out. And so I emailed Doug. I said, I want to, I'd love to see the incubator, at 953. And he actually didn't respond to me. And I was like, oh man, what's going on here? So I emailed him again. Can I see the incubator? Oh yeah, shoot. I've just been busy. Come by, Nick. Come by. And so, anyway, not to digress too much, but my wife also has a startup company, and she happened to have our car. We only had one car, and she was, gone that day. She was in San Francisco or something. So I get this other, this lab assistant from Mark Levine's lab, who's like wearing this like bright tie-dyed shirt, and I'm wearing like shorts and a t-shirt, whatever. And we go up and, we drive up and get in his like old, beater car and drive up to the 953 and jump out. Hey, Doug, what's going on? And he gives us a tour of 953 and he starts showing us all the equipment and stuff. And I was like, oh, that's so cool. You have all this, joint equipment that everyone can share. And wow, you only need one lab bench and you can have access to millions of dollars of equipment. This is fantastic. And he like looked at me, he's like, oh, you really drank the Kool-Aid, didn't you, Nick? And I was like, yeah, this is great. And so, we get through the whole thing and he's like, well, what are you doing? And I start, he's like, tell me what's your company? And I said, oh, it came out of UCSF. He's like, oh, you got really interested. And then so I talked through the science, and he told me later that he didn't quite understand all of the things. I was talking like 3 miles a minute, and he thought my enthusiasm was good, and Kayvon Showcat was good. So he figured, well, this seems pretty exciting. So at that moment, he said, well, what's your money situation? And I said, well, we have this term sheet. He's like, well, do you have the money in the bank? What's going on? And I said, no, not yet. And he said, well, would you be interested in another term sheet? And so that's how the whole thing, turned around. And then, MBC stepped in, Mission Bio Capital, and they've really supported us since then. And I think it's really the confidence of the team at Mission Bio Capital that's given like me the confidence to like push the science forward, saying that they put their trust in us to do the work. And so, it really gives us the confidence to say, okay, we can do this, even though we feel scared and like everything's not working. And then, you get to these points, but you have the confidence of these really talented and exceptional people to like help us to get the confidence to push it forward. It makes the whole thing, really, really fantastic and really fun experience. And I think a lot more fun than it would have been maybe working with the other venture capital company that we're talking to.
Wow, that's great. And that's a great example of how if you don't get an answer right away, to try again, to send that email a second time, because people miss things all the time. It's interesting you talk about the ID to IPO that you went through, and obviously the great experience at MBC BioLabs. I was curious if there are other entrepreneurial resources out there that you've taken advantage of that's helped your success.
Yeah, I mean, well, I guess like through MBC, we got hooked up with Carta, which is a great system for organizing cap tables, because I think that can cause a lot of issues for different companies. If you don't manage the cap table right, and then people's shares get messed up, and so all those kind of things, that's been a really big resource from the entrepreneurial side. But I actually think that just being at the MBC has been, I'm trying to think about external things, and I guess I'm having trouble thinking of those because of all the amazing resources that the Mission Bio Capital or MBC BioLabs offers. Where people come in and they say, they have guest speakers, from Promiga or other companies coming in and showing their best technologies, and then you get exposed to those different technologies and get a way to bring them in really quickly, or like lab organization technologies, or even just, like I said, talking to Doug, who's always thinking about how to optimize processes. I guess Doug's a singularly great resource for this kind of thing. I don't have any great outside one other than that, but.
No, that's great. That seems like a great network. We actually want to step back a bit real quick. I was curious, especially for our UCSF audience, I wanted to talk about what the process was like of patenting your work at UCSF, and then what was the licensing process to bring that out, and just in general, any key tips for people looking to spin out a company.
Yeah, no, I think the UCSF is actually a really exceptional place for doing that. So they joke in the Office of Technology transfer at UCSF that they have like an entire division just for Kayvon Showcat's technologies. So I guess it can be, I guess the experience might be different for other people, I guess I want to say, from other labs. But for me and for Kayvon, it was kind of a well-worn path of working on the patent. And so they actually supported us a ton in terms of the process of getting the license finalized. So they had, their lawyers, the UCSF lawyers, worked with me to write the patent up, the initial patent. And then, basically, get that out the door before the publication, so that we were, really covered in terms of that. And then, it was really a learning process for me and Dan. And so, the first thing we had to do was go in and actually license the technology, back from UCSF, so we could use it without, I mean, at the end of the day, none of the work would have been possible without UCSF. So it wasn't like, I think in some companies, they want to try to screw over the university or something like this, but we were, obviously happy to support UCSF and make sure the process would work both ways. And they were also really, great to work with. So we worked a lot with Ellen Katz, who's in office there, and she's just been a fantastic resource. She's done this, a bunch before, so she knows what's going on, and she's really no nonsense. So she doesn't beat around the bush. She really helps you out and tells you directly, okay, this is what you got to do. Here's how you got to get it done. And so that was really amazing. And they really worked with us on the licensee terms, which was, it was not, they knew we didn't have that much money, so they were willing to license it to us for not a huge, payout at the beginning. But then, they had ways of calling back the technology if they didn't feel like we were pushing it forward enough. And then they also had ways of changing those terms over time. So if we raised a certain amount of money, then the terms would change and these kind of things. And then actually one part of it was also that UCSF would have a position in the company, a sort of a non-dilutable position in the company that we were happy to work with them on. And they still have actually a position in the company right now. And I think they were even willing to put capital in the last time that we raised money. So, it was really fantastic just working with the office at UCSF and having them help to push it forward. It was never like an adversarial relationship, which is completely different than other universities. So yeah, I don't want to badmouth any other universities, but I know that it actually can be much more difficult at other places than it can be at UCSF.
Oh, that's great to hear. Curious how you talked about them still keeping a stake in the company, and how did you know when the right time was to start raising venture financing?
Yeah, I mean, that was really challenging too, because we, like I said, when you have a very, very small amount of money and nobody's willing to give you any more, you just like pinch every single penny, as much as you possibly can. And so, we went out to try to raise money in 2013. We went to all the VC shops that we could. And I think everyone was really gun shy at the time in terms of neurodegeneration and all the failures across the field. It's still littered with failures. And so what they really wanted to see was in vivo animal data. And so we said, okay, well, what in vivo animal model do you want to see this in? Because all of the drugs that have worked in all of the in vivo animal models have failed in patients. So like, I don't get it. Like, you want us to have it work in a model that's going to fail later on? They said yes. And we're like, doesn't make any sense. So we did the best we could to get to that goal, which took a couple of years. And so we really, I don't know, looking back on it now, I mean, right now there's so much money in venture capital and so much money in biotech that it seems weird that we couldn't go out there and raise money for what seemed like a good idea. But at the time it was very challenging, and I think, so like what we did was just try to build the best position we can. I would give a lot of credit to Dan DeRelay, who, when we were starting it up, he realized that, we both realized, we discussed, but he really, helped push it forward, was that, we had to get in and get our own IP and get it on new compounds to really build out that base. And so that was what, we decided to put our like really limited amount of capital to doing, was, to building out the patent profile for the company. So making a few molecules, working with really exceptional medicinal chemists. Actually, another story of reaching out was actually Bob DeVita, who's been our consulting medicinal chemist since Mitochondria started. And he saw Kayvon give a talk and literally just reached out to Kayvon and said, hey, if you're ever in the market for a medicinal chemist. And then Kayvon said, yeah, we are actually from Mitochondria, so come on and help us out. And so, and we're still working with him years later. And so it was really just to take these, he was a 25-year vet at Merck, and take his experience, design some new compounds, find the cheapest CRO we could that could make the compounds, and tell them not to rush anything, just, keep the prices low. And then, we drove up, I was in New York at the time, we went up to see CROs up in Boston and then outside of Boston and Worcester, and, get some simple cell-based assays going and just test the compounds with literally the bare bones budget we could possibly find, just to see if these new compounds would have any activity. And then, what our plan was to, develop that IP a bit more and then go back out and try to raise money. And so, it was in that process that we actually were then, as I mentioned, exposed to the VCs through Kayvon, where he had someone reach out to him, and, he knew kind of the work that we were doing, and we'd filed the next patent. So we had at least some more coverage. And that was kind of the moment when, we felt like it was a good idea to go back out to the VC. So we were also running low on money. So it was like, we have like $5 in the bank. That's always a good reason you should go to the VCs. Don't tell them that you're out of money. You say, oh, I have so much money, but you can give us a little bit more. Because if they know you don't have any money, then anyway. And so, it was really that moment where we're like, okay, we're running low on money, but we have the patents. We kind of did the things we set out to do with the initial like sub-series, sub seed, whatever you want to call it, funding. And then it's like, okay, well, we need more money to run the company. So that was a good time to go to VCs. But I think now it's completely different. Now it's like, I feel like professors just get approached and they say, okay, here's a check, just go start a company. It's like they don't even ask them what they're working on anymore. But anyway, I've heard, not for me, but for other, from some of the professors from UCSF, they're just like, go for it. So it might be a little different now. So maybe I'm not the best person to ask about that, but that's kind of how we did things. We just used the money we had to get to the next level and build it out and then get to the next step. And that's kind of the same thing we've done once we got the first financing from Mission Bio Capital, was just to build it to the next step and just take that amount of money they gave us and say, okay, what are end goals here with this amount of money, and get to that stage. And then don't try to solve all the problems at once. Just try to get to the next milestone that you can, that's deliverable with the amount of money that you have. And then actually just focus the science as much as you can. And again, I'll give credit to Dan DeRolay, our CEO, for this, where he's kept the company extremely lean on the administrative side. He's both the CEO and the HR director, and he does sort of everything for the company. But we were able to then put all the money back into the research and not have to spend additional money on overhead and other things that were less necessary. And actually, again, another plug for MBC. You can tell we're actually still there, so you can tell how much we love working there. But they were able to also supplement that with a lot of resources of helping us, like hazardous waste disposal and shipping and other things that would have been really challenging for us, but we were able to sort of lean on their resources for that as well. And so that helped us to not burn through too much capital too quickly.
Super cool, Nick. Maybe just going back to the real beginning of when you showed up at MBC BioLabs. Do you remember what your first set of experiments were? What were you really trying? What did you need the lab bench for?
Oh man, that was so tough. So Doug had a thing where if you did an experiment your first day, he'd give you a bottle of champagne.
Still do.
Unfortunately, we did not get. We were like, it was super challenging. The first set of experiments was just to replicate earlier experiments, and we just completely failed on that front. But it was really like to just go in and say, do we had this drug before, we have these new molecules we made. Let's go back and test them and just recapitulate the stuff that we've done before. Can we recapitulate, what we've done before? And then it was all of the new, publications that had come out since then. And it was just to say, okay, I haven't been doing research in the field for 5 years now. I've been working on this other project, or maybe 4 years, whatever it was. It's just to go back in there and just get things working again and really, order cell lines and recapitulate the biology. And we realized that, there's a reason why there's no other companies working on pink one. It's just a really, really difficult target. So we had a lot of failures up front. We had a lot of, the first experiments all failed miserably without any question. And so, we just tried to get things working, and that was really, where I think we developed the approach of like trying to get the big, huge difference, right? And never settling for just like some subtle 20% change, which I think, sometimes you can get like, coaxed into trying, where you think, oh, there's a subtle difference, maybe I can just like use that as my screen. And we were unwilling to settle for anything less than like a really huge difference between, basically adding the drug and seeing some difference in the cells and saying, expand your signal to noise window and really take it from being something that was like really an academic assay to being something where you could actually do a screen on it and find new compounds. And so that was our biggest goal, which is like make the science as robust as possible and never settle for anything that was like less than, very reproducible and very high signal to noise. So that was kind of like the first set of experiments, but the first ones all failed.
Once you got the rust off your nitrile gloves, what were the kind of key things in building that initial team? Any kind of input for people who are new to startups or thinking about starting a company, about how to kind of frame those first few hires and what to look for in people, and outside of scientists without rusty nitrile gloves, what do you think is some of the things they should be prioritizing?
No, that's a really good point. I think the first hires are really critical to building the company out. And unfortunately for small companies like ours, where we didn't have any real great publicity, it was probably, it was extremely challenging to go up against companies where, they were willing to pay, salaries, like over 6-figure salaries for jobs that we were not willing to, so you're competing against, basically companies that both have more resources and more publicity. And so it's really important that you get people in that, are dedicated and want to push the science forward and are also really, really careful scientists. And so I think that's the biggest thing, to focus on. And so our first hire was this guy, Chad, who still is at Miami County now. I think he's running Western Blots right now, probably. Basically he was at UCSF at the time. He was a postdoc at UCSF. And so, we were digging through all these mountains of different resumes. I don't want to say they weren't great. They just weren't maybe exactly what we were looking for. And when we came across Chad, we really, we said, wow, this looks like a really fantastic resume. We need to go meet this person and make sure he is who he says he is. And so we emailed him, and he said, okay, I booked out the whole auditorium in the Gladstone, at UCSF. We said, okay, let's go check it out. So we walk over there, and he brings us in this giant dark auditorium. He's just up there by himself, and he takes us through all his slides of all his postdoc work. And you can tell he's just so excited about the science and excited to push it forward, and really excited just about the potential for helping to cure a neurodegenerative disease. And he really had that drive, and he was willing to do what it took to get it to work. And that was really critical for early hire. And then I think as you build the team out, it's important also to know what the strengths and weaknesses are for different teammates. So not everyone should be, I think early on you want a lot of people to be really operating at a high level, but as you get further down, you want also some people who can who can be more specialized in terms of certain tasks. And I think that's also really important, because if you have too many cooks in the kitchen, it can be challenging to get anything done if everyone wants to design the experiments, but no one wants to actually do the experiments. That wasn't something that happened to us. This is more just sort of general feedback. And so I think that can be really important as well. And so finding like good RAs to come in and do certain experiments and help out to build the robustness of the data by just repeating things multiple times can really help out early on as well. And I think that's not a secret. And I think you'll find it, especially if you're trying to start a company now, it's actually a very challenging market for RAs right now, where they get offered just crazy amounts of money from large companies, where it's impossible for a small company to match that. So I think the only thing you can do to match that is to have hopefully really exciting science and be really passionate about it, but also to be willing to give up equity early on, to get the best people, because you're not going to be able to match them on the salary. And so if you can say, look, we'll give you options in the company, and you'll have a stake in the company, and that will make them more excited to join a smaller company, where maybe they're not going to get the same level of benefits that they get in a larger company, like not some huge target bonus each year and all this other stuff, and not a huge salary, but maybe if by giving them equity in the company early on, that can help to build the excitement in the company. I think sometimes founders are so focused with keeping all the equity that that could be a drawback.
That's super good advice. How big is the Mitochondrion team today? What's your headcount now?
I think we're I don't know, I think Doug might be listening. So but we're nearing 20. I don't want to say we're exactly, so I don't get kicked out of 953. But yeah, we're nearing the 20 person number.
That's super cool. So you must have learned a lot of lessons along the way about hiring and team fit. It must have been a tough process. Any resources that you used in that process? You have did you go to people that would help you recruit? Did you have any kind of hiring boards that you went to? What was kind of your strategy there for soliciting applications? You a big LinkedIn person? What was the strategy?
No, I only started using LinkedIn hiring recently, which actually is pretty great. We just use Indeed for all of the early hires. And I guess for me, it was really casting a wide net. And so one thing that I did was during all the hiring, I formulated a set of questions to ask all the different people we were trying to hire, because I felt like I had to find the hidden gold a little bit, not maybe the person with the stellar perfect resume wasn't gonna, maybe they'd apply but they wouldn't necessarily come, or maybe they were looking for a competing offer, so it was hard to hire those people. So it was kind of looking a little bit deeper and saying, okay, this person looks okay on paper, maybe I'll just pick up the phone and call them for half an hour and just see if they're really I don't want to say bad things, but a normal person or something, And so I would cast the net pretty wide, where I did a lot of failed interviews, where you get on the phone with someone and you realize, 5 minutes in, this is not going to work. But I was there, and I think we also found some scientists who maybe not as gregarious or as outgoing. And I think that can be hard, because at bigger companies, maybe it's easier for you to say, oh, I'm just going to get the person with the best resume, and they're really outgoing, they're willing to talk about themselves. But so by having a regimented series of questions to go through, I think you can get rid of some of those biases. So you can, you have the same questions. You can, you can write down all the answers, and you get a direct comparison between different applicants, as opposed to maybe one person is better at talking, so they talked to you for an hour, and you came away with feeling that they were the best applicant, and maybe someone didn't, and then you would, but in the end, that the person who didn't talk as much actually might be the much better person for the job or role that you're looking at. So we still ask those same questions to all the new people who apply to mitochondria. So doing that first 30-minute screener call and then bring people in. And I was probably also too willing to bring people in, as I'm sure people in mitochondria will tell you. There's a few people we brought in. We still talk about those interviews with some laughs. But yeah, I think those are the major resources. Was just trying to figure out the list of questions, and we didn't really ever use recruiters or official hiring websites or anything like that. So those are probably good ideas for other people to do, but we just didn't apply any of those lessons to mitochondria.
Gotcha. Thanks. That's super useful advice. One other kind of question, now that you've kind of gotten the science humming, a couple of years in, things are going really well, team's growing it at mitochondria. What kind of made you start thinking about engaging with strategics? When did you think was kind of the right time to start talking to people? Can you talk us through that process a little bit more for people who aren't so familiar with talking to pharma and kind of how to navigate through that?
No, I'm not familiar either. It was actually Peter Parker who told us that we had to. Peter Parker said, and before the end of the first year, you have to have a partnership in place. Oh crap! What the? So let's go and learn. Oh, crap! But I mean, let's go and learn how to do this. And so it was actually through the board, who really, Peter Parker really helped us to recognize the value in getting a strategic involved. And then we just went and learned about that. And fortunately, the person at AbbVie had already contacted us, before, around that time when she contacted us, Milagros. And so she saw, the science, and she was excited about it. And so, we were able to start discussions with her really early on. And we ultimately partnered with AbbVie. And it was, just through having those initial discussions and kind of listening to them and going back and forth and saying, okay, showing the science, and then saying, okay, you're not at the stage where we can start, continue this conversation right now. And so we'd ask okay, well, what would you want to see that would help us to push this forward? And then it was actually just keeping that relationship going for a number of years that really helped us out. So then, it was the next year she'd come back, hey, how's the science going? Okay, we did these experiments. Okay, this is looking better. We're still not ready to engage, call us back in a year. So then, that discussions kept going on until finally, they said, okay, now we're interested. Now I think, we're at the point where we can, push this to the next level. And I guess you kind of have to listen to them and hear, what they want to see, I guess, because there's I think for the kind of partnering that we did, it was you have about 500 companies, and then only one company will ultimately partner at the end. So they go through many deals, and then they get to some deals get advanced to the next stage, and these things are kind of slow. And I think overall for them, they like to see a lot of repetition, a lot of replicate experiments on doing the same thing over and over again and seeing the same results and feeling confident with the science. And then also they have a lot of other sort of things that we were not really used to doing, which actually I'm sure there's resources that you can use. I can list out all the different things that as a small company you should have if you really want to partner, but it's just having things as simple as SOPs and quality assurance documents and all these other things, and your data controls, where's the data, how do you know the data are unaltered, do you have the raw readout from the machine, and then you can trace the raw readout from the machine all the way to the Prism file where you're showing us a graph, and there's no time when you went in and deleted values that you didn't like or anything like this. So they like to see a lot of that kind of corporate diligence that we weren't really used to doing. We were used to just sprinting ahead as fast as we could, and obviously doing as good of science as we could, but we weren't writing formal reports. A lot of it was in PowerPoint. And so maybe if you wanted partner with a strategic, probably getting that stuff in order sooner. But obviously it didn't hurt us from getting the deal across the finish line. It just meant that we had to do that stuff sort of on the fly when they ask for these documents. Okay, where's your QA documents? You go Google QA documents. Okay, let's go write some QA documents down. And then we'll send those back to you in about a week. And it's here they are.
Oh man, that's a great story. A lot of people are skeptical about engaging with pharma at early stages. Tell me a little bit about that process. A lot of people are worried they're going to behave badly, or think that the biotech is kind of good enough on its own. How was that dynamic? Was it a mutual benefit? You learned some stuff from them, and they learned some stuff from you, and how did you feel about those relationships when you were pretty early?
Yeah, no, I think that's always the worry, right? Is that they're going to just come in and learn everything that you do and then feel it. And so that was definitely something where we were, really cautious in the beginning of showing them any sort of confidential data, without having these really careful engagement letters and everything else, so that in case that did happen, that we were protected from that happening. Because it does feel a little bit like it's the sort of evil empires coming in to steal everything that you have. But I think that also just really that also really depends on the group that you're talking to. And so ultimately we chose to partner with AbbVie because they put science first, and it really from like the top down at the company, it's really all about the science, and it's all about how you can actually help patients, and that's like the most important thing. And then the way to get to there is to have really, really good science and always follow the science, and never sort of follow what politically might be exciting at the time or other things, but really follow the science and just stay with the data. And that was what really brought us to that collaboration. And I think other groups also maybe did the same thing, but maybe in just slightly different ways. And so it really was about a personality match. And I'll give you other advice too. If you ever do get into one of these things, and we got this from the board, from Doug and from Peter and everybody on the board, but it was really like, get multiple competitive offers, because they will just rake you over the coals if you don't have any competitive offer. If they're negotiating against themselves, there's really no reason for them to do that. But if anyone else comes in with an offer, then you get them to kind of play off each other. But that's probably can be said for many things in life, I guess.
That's super good advice. I just wanted to pause for a second and say to the audience watching it, if you could just, if you're interested in any other questions, we're going to have a few minutes at the end to ask Nick some audience questions. So please put them in the QA or in the chat. Either way, they come to the panelists. I know Jenny has a couple other questions to wrap up before we go to audience questions, but Nick, thanks. That was super helpful.
Yeah, sure. I guess just in general, any reflections on entrepreneurship that you'd want to share to graduate students, postdocs, even faculty to make the leap? I don't know, is there anything you wish you knew when you started the journey?
Yes, I think the most important thing is to find someone who knows the business side as well, because I think that's really critical, that you have someone who can take on a lot of those aspects. Because then, let's just say you're a young faculty at UCSF or a grad student or a postdoc, that'll allow you to really focus in on the science part of things, and not spend all your time I don't know, figuring out all the other kind of documents and other things that have to be done to make sure a company is successful. So that's really critical. And that's, to find that person to work with, is really important early on. And that it just frees you up to focus on the science and not get bogged down by all those other sort of things that can happen in early companies.
Great. So is that like a co-founder, or if so, how did you find your co-founder?
Yeah, no, I think, yeah, exactly. Like a co-founder, a set of co-founders to go in, early. And it's probably better to have someone who has experience actually doing biotech companies, because there are a lot of sort of nuanced things with biotech companies that probably other companies don't have. Yeah, so my co-founder is actually also my brother-in-law, and so we found him, through that connection. And it ended up, it's sometimes creates a little strain, but overall, I think Doug and Peter were happy, because they said, we know that you guys always have to go have Thanksgiving together, so you're never going to be like hate each other. It's like you always have to kind of work things out. And I think that's the case, for us, as well. That at the end you have to work things out. And I mean, for any co-founder, you have to work things out. But I think that can be there. But probably there's also drawbacks to doing that. So I wouldn't necessarily recommend that. But I think it's really about finding the person who's right at the time. And if you have someone who's like clamoring to be your co-founder, and has an excitement to drive the science forward and let the science really speak for itself, and it doesn't have a huge ego, I think that can be a really good relationship.
Great. So we have a question in the Q&A, and I'm not gonna lie, I get this question all the time from students at UCSF, so it's perfect. This person asks, when is the right time basically to bring it from academia to start a company? Do you have to have accumulated enough data to start the company, such as good in vivo, or what is that turning point?
Yeah, if you want, I mean, that's actually a great point. Having good in vivo data is actually a really good point. I think, but it really matters what kind of, I guess that's a really good, okay, if you're making a therapeutic company and you have a certain disease, having in vivo data is probably, almost critical, could be critical. But I think for more of like a platform type company, where you're going to use some technology you're already using in academia and then apply that to multiple different projects, it can be very different for each of those. So I think it actually probably the best thing is that you just go out there as early as possible and, pitch your idea and get your elevator pitch, good to go, and, have a partial business plan, have some sort of IP protection around your idea, and then go out and test it on people, go to bars and go to restaurants and just talk to people and say, hey, does it seem like a good idea? And if they have no idea what the hell you're talking about, then you should probably refine your pitch to where you can literally get the pitch off off your tongue in 15 seconds, because that's what it's going to take. It's going to take you going out there and pitching and getting told no over and over and over and over and over again. And each time you get told no, figuring out why did they get told no and how can I improve that, for the next time. And so I think as early as possible, trying to refine your idea into a few key points, into a few key slides, and then really just practice pitching it and practice selling it is probably the first thing you should do before you're even ready to that's even before you're ready to go out and raise the money, right? And then once you kind of have that somewhat aligned, then you want to put together the right data package. But you can almost learn that by going out and pitching it to different potential investors and getting an idea of what they want to see, right? So maybe for your oncology project, they need to see complete remission of tumors in these mice under these conditions. And you can kind of go back to the lab and say, okay, well, I can either work on getting those data, or I can know that that's the first thing that I should do when I start the company, and then I can raise the money to go answer that question. But you can kind of a little bit learn from your audience, as long as you keep that core part, the core idea, which you've come up with yourself and you've really refined, and that's really what will guide the company forward. But then the rest of it you can kind of figure out as you talk with different people, I think.
Yeah, we often tell people to do a lot of customer discovery, and just the more people you can talk to, the better. Another question we received was, do you have examples of trusting your coworkers and letting go of some parts of the business and the research, going from your project to a team?
Yeah, no, that's definitely extremely challenging. I think I think professors have trouble doing that as well. I talked to some professors who still, they go through and analyze all the data sets themselves because, they either don't have trust, or they feel like they know they can just do it better. And so they still do those different aspects. And I think that's a huge part of starting a company, is actually, letting other people have your trust that they can go in and do the analysis themselves and do the experiments themselves. And I mean, maybe talk to people in my company, I think they still tell you that I have trouble with that sometimes, where I have trouble letting go of certain experiments that I want to still help out with, or I ask them to take videos of what they're doing so I can help them to feedback on it. But that's absolutely the case. And I think it's really challenging because you have to let go in a way that you don't know how it's going to work out. And if it's a really critical path thing for the company, you still have to let go of it and have that trust that it'll happen. And sometimes it's really about taking that first step and just saying, all right, I'm going to let go completely and let's just see what happens. And then you get see the results back, and just try to analyze them together. And it's really about learning how to mentor someone else to do an experiment versus doing it yourself, which maybe professors wouldn't have that issue, but for postdocs or grad students, it can be difficult, right? To go in and say, I'm just going to give up what I am the best at doing probably in the world, and I'm going to let someone else try to do it. But you really have to do that and just say let it go.
Great. And so, and one last question, I guess, where do you see yourself in a few years?
Hopefully curing Parkinson's disease. But I personally, I don't know. I mean, I've been trying to think about this myself, but I'm so busy right now trying to get this company to the next step that I haven't really thought that much about it. But I guess once I started Mitochondrion, then I got really excited about starting the next company also. And so it's really I think I feel like I've learned so much from this experience that for me to go out there and take another idea from beginning to end would be really, really fun. So in a couple of years, I don't know, I could be either continuing to push Mitochondrion work into the clinic or starting the next startup company, or maybe I'll go back to academia. I'll finally be a professor, and my dad will be happy. I can go back and get the Nobel Prize finally.
Well, that's great. I'm sure whatever you decide to do will be amazing. We have another question here. How do you maintain creativity or explore new avenues in your research? One drawback of biotech or later stage startups is to making sure the avenue you choose is profitable, how do you determine if it's worthy enough to pursue?
Yeah, no, that's a really good question. That's probably the reason that a lot of people who are more on the pure academic side don't want to go to companies, because they don't want to feel that restriction on their research, and they don't want to feel like someone's telling them what they need to do because they need to be profitable. And so again, I don't have a great answer because I didn't experience that, because it was probably different for the approach we were taking against a disease that afflicts millions of people and has no cure. So I guess it was always obvious that if it worked, it would be profitable. So we didn't have to make that sort of, we didn't have to defend that part of the research. But I think, inside the company, we do sometimes do side projects or things that are not directly related to, exactly where the company is going, and are more sort of avenues that we want to explore. And I think there is room for creativity, in a company. Probably as you get bigger, there's probably more room for that kind of creativity in terms of figuring out, like, sort of, I'm just go back to this question. How do you maintain creativity or explore new avenues? Okay, actually, I have a better answer now. Now that I thought about it for a second. So I guess, overall, I think, people feel like it can be restricted. And so another way that we actually went back and tried to address that, or I did, was that we kept really close connection with a lot of academic researchers. So we have a really excellent SAB, and I basically serve on panels with basically a lot of these academics. So through Michael J. Fox Foundation, I was on a workshop last week with 15 of the top people in the world studying pink lung and Parkin, right? And being able to talk to them and see what they're doing versus what we're trying to do. So I was trying to keep I was the moderator for one session, and so I was trying to keep the conversations focused on what in vivo models can I test with my drugs to cure Parkinson's, right? And then we got on this conversation for like 30 minutes where they were just talking about all the new hypotheses in the field. And I got completely swamped because these are like extremely famous professors. And I was just like, okay, you guys just talk. But you get to hear what's like really, the critical question in the field, and what's like the next step of what they're doing, because they're always trying to do things that are, 10 years out. And, in a company, you're kind of trying to do things that are at this stage. So by keeping that close connection with researchers, allows us to kind of keep those like lanes of creative freedom open. And like, so for example, there's a new finding that we had, which we thought, this is really cool, but we don't have the bandwidth to pursue this at Mitochondria. And so then I went to Michael J. Fox, and I talked to the people there, and I said, you should totally present that to all of our academics and just basically say, you guys should take this. We have like a nice compelling data package. And so we can almost like, we don't get to do the creative work ourselves, but we can like help at least advise and learn from the academics doing the creative work. And then we can also like pursue, co-projects with them, or give them our drug, and then they can get publications with it, and we can help them design the experiment. So by keeping that like, the crosstalk open, I think that can help to keep the creativity, flowing, even if you feel like you're kind of doing things that are a little more consigned to, to what the direction of the company, I guess.
Great, and I think we have time for maybe one last question, briefly. What books do you read, and other activities do you keep your creative mind flowing? Short.
That was great. That was a good question. I'm actually reading the Stormlight Archives right now. I don't know if you know these, by Brandon Sanderson. It's really cool. It's like, I love it because it's this whole other world where a lot of the natural laws are the same, but he's done a really good job of creating like new magical laws that are like, but very defined within the universe. And he's very consistent with all of it. So like my scientific mind likes that, because it's not like, okay, everyone just flies. It's like, oh, here's why they can fly, and here's how they can fly, and here's the limitations of them flying. And it's not, so that's really fun. I love to read those books, and so I'm sort of in the middle of that right now. And then the other thing, the most important thing I think for me is really to keep active, and like running and swimming and biking and surfing. And so, I do all of those things. And so in the fall, oftentimes like between meetings, I'd run out and get a quick surf session in, then just work like later at night or something to sort of make up for the time, when the surf was really good. It's like, I gotta go surf. There's no question. You just gotta go surf. So I think keeping, having those activities that are really like, essential for you to, keep your focus and keep working hard. You don't want to forget that, because otherwise the stress will just like kind of overwhelm you, and you won't be able to keep being successful,
Yeah, I think that's really important advice for all of us, especially in COVID times.
Well, thanks so much, Nick. This has been super great. We so appreciate your time. This was super enlightening for me personally, and I bet for our audience from UCSF and MBC BioLabs. Thank you so much, Jenny, too, for co-hosting this today. This was a lot of fun.
Yes, thank you so much, Nick.
Thank
Some
you.
more links in the chat if anybody wants to check out links to MBC BioLabs or learn more about the UCSF Catalyst program or UCSF Launchpad. We really appreciate everybody's time, and Nick, that was fantastic. Thanks for sharing your startup wisdom with all of us.
Okay, thanks so much, guys. Thanks, Jenny. Thanks, Zach. And if anyone wants to learn more about mitochondria, you can go to our website or connect on LinkedIn if you have any other questions. So thanks.
Thanks, everyone. Bye now.